Drug developers, investors, researchers, and corporate executives wrestle weekly to understand what is happening in commercial development of NASH medications. Join hepatology researcher and key opinion leader Stephen Harrison, C-suite veteran Peter Traber, and forecasting and pricing guru Roger Green as they discuss the issues affecting the evolving NASH market from their own unique perspectives on this week's edition of Surfing the NASH Tsunami.
For everyone with an interest in NASH or more broadly fatty liver disease, surf's up because episode 23, day 3 of the first Digital ILC is starting now. First, I want to thank you, our listeners. I mentioned yesterday that the uptake on day 1 in the first 12 hours was the fastest we'd had with any episode to date. That record lasted exactly a day because the uptake on yesterday's episode was faster. So thanks everybody for paying attention. I want to thank in person, I'm going to name him, Ian Rowe, who sent a fantastic comment back that is now sitting on the discussion group for anybody who wants to hear it, and that I will mention probably to lead off our session next week. Also, we will be, I don't know what time we'll be dropping our podcast today, but since I hope nobody is, at least nobody in North America is planning on waking up early tomorrow because there is no Sunday session. It will be there by the time you wake up if you're in Europe. It will be there by the time you wake up in the US. Not sure exactly what time, but early enough. Okay. Basically, what we've heard from Ian and then from more casual comments is that folks are finding this a fascinating meeting. Lots of topics, lots of interest, lots of different directions. I'm going to go back to what Donna said on day 1 about the nice thing about her having been trained as an attorney is she can wait till the end of the conference to put the pieces together. By now, we should be in a position to start doing that. So what I would like to do for a kickoff is I would like to ask everybody the same question I asked yesterday. One thing you heard in the last 24 hours, and if not that over the life of the conference, that has you thinking differently about the challenges surrounding fatty liver disease than you thought when we started down this path on Thursday. Brave one, go first.
This is Donna. There was one quote in particular that stood out for me and, will be very important for the Global Liver Institute to focus on addressing. And it's that individuals who are obese face 150% greater medical spend compared to those who are non-obese. And so I think that if we're going to have data-driven policy, which I saw quite a bit of during the conference, these are the type of data that are the foundation of effective advocacy. And when we talk about, awareness, it's often so, fluffy or soft or broad. It's really about using statistics like this to bring these issues home to people and to policymakers to cause change. So that's one of the things that stood out for me today.
This is Jörn. I'm going to go second after Donna and mention an abstract I heard today in the NAFLD experimental session, which I thought was amazing because it dealt with sarcopenia in patients with non-alcoholic fatty liver disease. And while even in my head, I'm thinking of those patients as being metabolically impaired and being sarcopenic. Actually, the authors highlighted that they're not sarcopenic at all, but rather exhibit something you'd consider as myosteatosis. So that reminds me again that it's a multi-organ, multi-system disease, and it's not necessarily just the loss or the lack of muscle. That wasn't even there in this particular study. They used animal models to replicate that from the association in humans. But it's changes in the tissue that is associated with a more severe phenotype.
So Jörn, my apologies. I was remiss in not introducing you up front. I've been so concerned about getting the session in on time that I didn't. I'd like to welcome our guest today, Joern Schattenberg. And Jörn, having given that really fantastic statement, could you take a minute and tell everyone who's listening a little bit about yourself and what you do in your involvement in the community and the various roles that you play?
Yeah, thanks, Roger, and sorry for getting ahead of you there. And yeah, my name is Joern Schattenberg. I'm the director of the Metabolic Liver Research Center in Mainz, that's in Germany, and located in Germany. And I'm a gastroenterologist, hepatologist with a special interest on the studying of pathophysiological mechanisms of patients with non-alcoholic fatty liver disease, the development of novel diagnostic and prognostic tools, I think, and therapy, of course. And I'd really like to, thank you for inviting me to this very special and interesting podcast here. Happy to serve with you guys this afternoon.
And let me just add, Roger, in addition to being a world-renowned hepatologist with a multifaceted focus across a wide range of liver disease, he's tied for probably one of the top nicest hepatologists on the planet with Jean-Francois Dufour. So hats off to you, Joern. Thanks for joining us today.
Keep going, Stephen. I just meant about the thing that you took out of the conference.
For me, I'm just looking back on these 3 days of drinking a lot of extra coffee and, waking up like I did in the Army to be a part of this. And I think the recurrent theme for me that hits every day is the amount of data, the amount of interest, the amount of queries, the amount of questions that come relative to non-invasive testing, really for the 3 different contexts of use that we're looking for. That is the diagnosis of NASH from simple fatty liver, therapeutic markers of efficacy, and long-term patient outcome measures. And we heard more of that today, not so much in presentations, but certainly in the questions that were asked relative to the therapies that are being developed.
Very good. Peter, Louise, either one of you go ahead.
So I wanted to comment a bit on the meeting as a virtual meeting. I really made a 180-degree turn since the beginning. I was quite trepidatious about a digital meeting. I thought it would be problematic, but EASL did a wonderful job in setting this up and putting together the programs. And I found that there were some additional benefits to a digital meeting that I really hadn't thought about. For instance, one can do research on various topics on the fly while you're listening to the presentation. You can communicate with other colleagues about presentations who ordinarily wouldn't go to liver meetings, but might be experts in certain areas like genome-wide sequencing, which was a very nice paper in the late breakers this morning. So I felt that it was a very rich environment sitting in front of a couple of large screens and listening and interacting. And finally, I think the questions were generally greater in number and better than at the in-person meeting, because I think a lot of people who might be reticent to ask questions in a large group ask their questions. And we've all been in meetings where the same individuals get up time and time again to ask questions. By the way, I've been guilty of that, so I apologize. But I think we see a more heterogeneous population of people that are asking questions. So I found a lot of positives in the virtual digital meeting.
Louise, go ahead.
Yeah, Donna was obviously in the same session that I was in because, as you know, I have a connection with Australia, and one of the shocking statistics that came out of there was that 87.7 billion in additional direct and indirect costs to Australia accumulated across 10 years to 2025 from obesity is predicted. Now, this is a country that does not have Medicare covering FibroScan. So anybody who gets access to a FibroScan is only realistically once they've been sent to one of the major liver units. So they're predominantly diagnosing on ultrasound and liver function. And I think we have seen repeatedly through the conference over the last 3 days that liver function tests leave an awful lot to be desired. and the use of non-invasive technology is something that's been heavily underlined, whether or not it's direct access to GPs or within specialist centres, but basically racking up the amount that we do. So it'll be interesting to see where that goes in the future, because there was a very good article out by Leon Adams, and a lot of the major consultants in Australia last year predicting an 85% rise by 2030 in most severe liver diseases, particularly liver cancer and cirrhosis. So it's going to be very interesting to see where Australia goes if it stays out of the main fold on its diagnostic techniques.
That's interesting. That is really interesting. I think I have a different understanding of the dynamics of the economics of the disease and market development than I did 3 days ago. The papers about the lack of primary care engagement with obesity and the inability to translate from an obese patient to somebody who should be tested to liver, and the impact of things like the Wales experiment that was yesterday. And the ease of using some simple tests to figure out at least who should get screened into the process, and the improving quality of the non-invasive testing, all of it tells me together that the focus on making this more a primary care disease, or at least making all the initial diagnostic screens primary care screens faster, as quickly as possible, would, I think, benefit everybody. It would benefit patients. It would benefit the economics of the system. It would benefit our ability to develop solutions that work. So I think that's what I've taken out of this as much as anything else, is the importance of going back upstream to primary care to educate them about where the liver fits into everything else that they
As
look.
Brian Harvey said, I want to, support my board member and his, insights into the field, and to make sure that there's clarity around what he was saying, which is basically, what you have now, come to see the light, Roger, that and it wasn't meant to take away from the job that hepatologists and gastroenterologists do for severe patients, but unless we address this and all of the factors earlier on in primary care, we're going to ultimately be fighting a losing battle.
And just not to digress any more than I have to, you know what, I won't digress at all. There's a flaw in how he looks at that argument, which is what happens after you make the statement that you made, but we can come to that a different week. Certainly not today. We got plenty to do today.
Okay.
So what we will do with the rest of the day is the same thing we've been doing, which is hopefully a little more efficiently, which is we will go in order through the papers that we said, or presentations that we said we were going to discuss. See if we can take roughly 5 minutes on each one. They were running maybe 6 or 7 yesterday, a little bit of discussion after each, and then we'll go on to the next. In the order that it popped up quasi-randomly on the website. So on the website, actually, the first person that pops up today is Donna. And so Donna, you want to kick us off, please?
Well, I'm happy to. And I particularly have a wild card draw because when I originally set out to say which ones I was going to cover, I picked one on Thursday and 2 on Friday. So my Saturday was open really to what I found interesting, and also what occurred after dawn in the United States. So I want to give credit to Louise's nursing panel and to all the nurses in GI and hepatology, because I know what goes into partnering with a society to make something like their nursing partnership and track happen. But I spent a lot of time in the COVID-19 panel, really searching for answers, because the number one questions, whether from NASH patients or otherwise right now, coming to the Global Liver Institute are, what is my chance of getting infected for COVID-19? What would happen if I become infected? And and what are happening both with COVID liver patients who contract COVID, but also patients with COVID and how do their livers functions. And I think that the COVID-19 and liver disease panel did a really good job of addressing just about all of those questions, even if the final answers were, in some cases, it is still uncertain. And I certainly want to give a shout out to the secure cirrhosis and COVID HEP registries for the work that they're doing, because one of the issues that coming from a larger research background or a research engagement outside of, inclusive of, but outside of liver disease, my concern has been that there are not enough liver patients and not enough autoimmune patients in the general COVID-19 trials, and that is what they were seeing. So to have liver-specific trials that are really casting light on such things as the impact of NASH and obesity on having a greater severity of disease, looking at cirrhosis patients who are dying at younger ages than the non-cirrhosis patients, looking at rates of for those who do have cirrhosis, their rates of decompensation accelerating with COVID, and the more advanced their liver disease, the greater the severity of their complications with COVID, and so that that is so impactful as we design policies and safe care for NASH patients, and particularly NASH patients with cirrhosis, and give them the information that they need to navigate this. They also did spend some time on transplantation, and we know that NASH is going to soon become, or in many places is already, the #1 cause of liver transplantation. And so, we've been monitoring from the beginning, and I've personally interceded with both CDC and the American Society for Transplantation to make informed decisions about closing down and reopening and having access to transplantation for patients during this time. And so they laid out a very nice model, which is, I think, more guidance than has been given to date for centers who are in COVID and non-COVID, high prevalence areas, to understand how to reopen their transplant facilities and for whom. And so having COVID, thinking through COVID and NASH, which is what we do every day, and particularly in our COVID-19 response program. What this meeting has told me, and what this session today has told me, is that we need to continue a dual strategy of ensuring that there are NASH patients, there are liver patients with a variety of chronic liver diseases. There are autoimmune patients in these general COVID trials, and not just of hospitalized patients, but in trials of medical workers, of other frontline care providers, whether it's teachers or otherwise, both for treatments and for vaccines, so that when things come out, we know if they will work for our patients. And then the ability to continue to support liver-specific registries to develop that information is more important than ever. And I am so grateful as a patient for the researchers who have stood this up so quickly and focused on this, and just make a commitment to use this information as quickly as possible to help guide patients' everyday decision making.
So, Donna, since that was also my topic for the day, I'm going to piggyback a little bit and take about a minute or minute and a half and go from a slightly different direction. First of all, I agree with that. I was tremendously impressed at how many patient cases they were able to stand up for some of these analysis. The work from Oxford with 700 patients out of 2,000 who started in the screen, and what they were able to look at there was, I thought just tremendous, and agree the number of transplant cases as well. So things worth noting. As you mentioned, the more severe the degree of cirrhosis, the younger a patient is likely to die. Chronic liver disease didn't seem to be a lot different from the population at large in terms of age of death, but cirrhosis clearly made it younger. One of the things I thought was interesting was that 22, I think was the number, percentage of the cirrhosis patients at time of initial appearance in the hospital had no respiratory symptoms, yet respiratory was the leading cause of death for 62% of those people. So the simple fact that you went in with lungs clean didn't mean that your lungs were going to stay clean. The rate at which the severity of cirrhosis damaged the chances of survival, I thought were really pretty breathtaking. So there was that whole set of facts that I thought was important. And then the idea in transplantation about the trade-off between immunosuppression and the benefit of immunosuppression versus non, and dexamethasone versus prednisone, and the idea that patients who had livers longer, immunosuppression wasn't the issue, it wasn't a significant issue to be dealt with, it was just dealing with the disease, whereas people who were transplanted more recently, there was a much greater cause of concern. All that made sense, but the clarity around it, which I'm not doing justice, I thought was really pretty spectacular. And I agree, I was very impressed by what they were able to put together on that subject and how quickly. Other questions, comments? All right. Then Louise, I think you were next, actually, on today's list. Yes, you are. So go ahead.
Well, I will be a lot quicker than I was yesterday because I was doing a poster that was presented in one of the sessions, and it was the Varicine algorithm using successively platelets, liver stiffness, and INR improves and secures the screening of esophageal varices needing treatment. And I think I've mentioned earlier in one of the podcasts that the Baveno 7 criteria has been delayed by 12 months because of COVID. And I think obviously telemedicine has taken leaps forward, but there's a lot of work being done on trying to get endoscopy lists up and running. So obviously we use Baveno 6 criteria, and we've been using enhanced criteria in lots of institutions. So this was a look to basically say two things. What's good and currently available, which current proposal offers the best combination of platelets and liver stiffness value, and can the current best proposal be improved? So it was by Dr. Raviglione and the team. So from various institutions. So what they did was they looked at all of the studies with Baveno 6 criteria, anticipate, expanded Baveno 6, and they reviewed them for varices that needed treating and also those that were spared having endoscopy to look at how safe these procedures were. And what they did was they created their own algorithm, which they had, and they added in something called PLER, by which they just used platelets and liver stiffness ratio. And then depending on that score, being above or less than 17, so if it was less than 17, you went into a further review on something that was called PLEAS. Now that was by adjusted the PLER on etiology, sex, and INR. And what they were trying to do was avoid all of the scans that endoscopies that should have been done and varices were missed. So what they basically found out was that the enhanced Baveno criteria proved to be unsafe and unsatisfactory in both of missed varices that needed treating in both groups. So their populations, they did allocate out 2 to 1, various different liver diseases. So they did a good randomization of that. And it was 2,300 patients used. So by using their algorithm, they plotted that they would get an enhanced rate and they would miss less of varices that needed treating. But also Baveno 6 criteria came out high, and also the criteria that was discussed with Anticipate. So their poster contained a link to their algorithm, which I did try, but I did have to say I struggled despite putting in some pretty low figures for platelets and high liver stiffness measurement that seemed to come out okay. But when I did use the PLER, it did identify that the patient probably needed more work, but it had a good negative predictive value, or reasonable at around about 75–80%, but its positive predictive value when it comes out is very low. So I can't recall how low it was, but it was actually surprisingly low. So they've got a bit more work to do on it, but I think it did highlight weaknesses in expanded Baveno criteria, even if we're going to try and shorten endoscopy lists. So obviously using liver stiffness measurements, FibroScan can help us get down those lists, and I think that's where a lot of centres are going. But being able to look at different criteria as well, Anticipate, may be another one rather than the expanded Baveno criteria. So that was my paper today, which has some good promise and a little bit of work needed.
Louise, I didn't see that presentation, and maybe I'm a little confused. Can you clarify for me? You know, we have the criteria with FibroScan, liver stiffness, and platelet count to define the risk for varices and the need to do endoscopy or not do endoscopy. How is this different? Can you clarify that for me?
They used a platelet count of 402 GI or 2 liver stiffness measurements of less than 9 kilopascals. So they used a different level of kilopascals within their algorithm for VariScreen. If your platelets were less than 402 and your liver stiffness measurement was greater than 9, you then had their additional ratio of platelets to liver stiffness. If it was greater than 17, it proved there was a 0% chance of missing out on the varices. If you had less than 17, you then went on to their next level of their algorithm, which was the PLEASE one, where they added in, it was adjusted then for etiology, sex, and the INR of the patient. Now, their cutoffs were greater than 6.2. So if it was less than that and it was negative, then it had less than a 5% chance of missing varices that needed treating. And if it was greater than that, it was positive and was a greater risk. So it was interesting. But when I tried it, it was a little bit difficult to work through, and it did successfully put me through eventually. And so it's a little bit crude, but it certainly has promise. But it was the weakness that they defined in the expanded Baveno 6 criteria that I suppose surprised me, because that wasn't what came out of the initial data. It was quite strong. So it needs some more work, and I doubt people will be moving and stopping using the enhanced Baveno if they need to currently during this post-COVID period.
Yeah, just pragmatically thinking about that, at least in the US and maybe more specifically in Texas, I am part of a large GI practice with, we do, maybe 100 endoscopies a day. And when I would apply these criteria, it tended to be like only accepted if it validated the reason to do the endoscopy, because most of these patients are going to get scoped just on the risk factor of having cirrhosis, right? So they're going to get HCC screening, and they're going to undergo an EGD. And if you can make them over 50 and no prior scope, then they're going to get what we call a rotisserie, right? They're going to get an upper and lower. And and so it's not really used that much. Where it is helpful is kind of just reinforcing the fact that this patient is at high risk, and we need to make sure we don't let this one slip through the cracks, so to speak. But unfortunately, I don't see that being used a lot with the current payer system that we have in the US, at least in the private sector.
And I'm going to chime in on this and comment from the European side. I think it's a similar pattern we're seeing. You know, I don't get referrals for positive Baveno criteria for an EGD. So I do send these patients myself to be scoped.
Okay. Other thoughts on that one? So we will now move into the section of today's conversation that are about late breaker papers. Peter first, Stephen second, Joern third, and then if time permits, I'll talk a little more about the COVID and the liver section. Peter, why don't you go ahead?
Okay, I will try to be concise. The paper that I listened to, the presentation was on the ATLAS trial, which was entitled Safety and Efficacy of Combination Therapies, including Cilofexor, Percocet in patients with bridging fibrosis and cirrhosis due to NASH. And this is the result of the phase 2b ATLAS trial. It was presented by Rohit Lumba and has a multiple number of luminaries on in the authorship. I will say I've been waiting for seeing the presentation of these results since the announcement late last year of the trial results in a press release. This is an ambitious trial of 7 different arms with 392 patients, randomized 2 to 1 to 1, meaning combination had two times, and either monotherapy or placebo was 1. So there was a placebo group, a Cilofexor and Percocet and Selonsertib alone, and then those 3 in combination. So 7 groups. One of the kind of important things about this present study was that they had over 50% patients with F4 or cirrhosis. So they had NASH. It was evenly split, just about a little bit more NASH cirrhosis than stage 3 fibrosis. So it was a kind of a combination of late pre-cirrhotic and cirrhotic NASH. They had their endpoints were multiple, but it was based primarily on a paired biopsy that was done 48 weeks after therapy. So, excuse me, therapy for 48 weeks with a biopsy at the end and baseline. Now, when this trial was presented, it was in a press release. It was really said, you know, this was a negative trial. There weren't any significant differences in any of the groups, but they hinted that there were some endpoints that might be positive. And again, they showed the original data, which was that a 1-stage improvement in fibrosis without worsening of NASH. The only thing that was numerically different was the combination of Cilofexor and Percocet at 21% response related to a placebo of 11, but it was not significant, but it approached something that looked like there might be an effect. And they showed differences across kind of the histologic measures. But the interesting thing about this abstract is that they went on to evaluate the biopsies using machine learning assessment. And this is kind of an approach that I think a lot of people are taking now, doing a retrospective analysis of biopsies using machine learning, even if it wasn't a priori planned in the clinical trial. And they saw some very interesting results. First of all, the fibrosis effect for a one-stage reduction in fibrosis was now statistically significant for the combination of cytoflexor and fricosistat. And the other thing that I thought was quite interesting is that they saw an improvement in ductular reaction, which is proliferation of the bile ducts and seems to be quite important as a component of NASH, only recently kind of focused on, and certainly not included in the original pathologic classification of NASH. So in the end, they saw significance in other secondary markers such as KLT, bile acids, CK18, all most prominent with cytoflexor and fricosistat. One of the interesting things about this study is that, of course, cytoflexor is an FXR agonist, and as we've seen in this meeting repeatedly, the AE of pruritus is something that's quite consistent with the FXR agonist. And this brings up something that Stephen mentioned, I think, yesterday, and that is that we may, when we start doing combinations, be able to use lower doses of FXR agonists and not have quite the incidence of pruritus. And that's exactly what happened here in this Atlas trial. Their incidence of pruritus was actually lower than what we've seen in other trials, which are around 50% at the high doses. But here they were half of that, and it's because probably they were able to use a lower dose of cytoflexor. So a very interesting trial. Nothing extraordinary that came out of this trial, but I think it showed a couple things. One, the ability to use machine learning on biopsies to gain additional information and maybe even show significant differences in your trial. I would hasten to add that all the data we have on prognosis of and being related to fibrosis stage is related to a pathologist reading and not machine reading. So there is not a direct read-through of the fact that there's significant difference with machine learning read and not a significant difference with a pathologist read. But we learn a lot more information, such as the ductular hyperplasia. And then the other thing I think is the potential for reducing adverse effects such as pruritus in combination therapy by enabling lower doses. So an interesting trial. I'm sure that, I'm not sure actually that Gilead is following this up in a phase 3 program. Maybe Stephen or Joern know. But I think this was an important paper at this conference.
So I found it interesting as well. I thought, at the end of the day, this was a pretty robust phase 2B trial, and there were 7 arms, I think, and so there ultimately some of the numbers weren't maybe as robust as we would like them to be in a 1-year phase 2B trial, despite the overall total number being almost 400 patients. So I think one of the comments that Rohit made when comparing the dual FXR ACC arm with placebo, you had a placebo-adjusted improvement in fibrosis of 11%, 21 versus 11, or 10%.
Yeah.
And that did not reach statistical significance, and it could be because there just weren't enough patients enrolled to show it, or maybe they didn't treat long enough. The other thing that I thought was worth noting, I didn't, and maybe you can comment, I didn't see necessarily a breakdown between the F4s alone and the F3s. Did they break that down?
Very good point. No, very good point, Stephen. They did not. And I think that both that and any two-stage reductions would have been useful to see as well. But no, they didn't break down cirrhosis versus non-cirrhotic NASH. And I think, one of the things is that the numbers are amazingly close to the phase 3 numbers for the Regenerate trial, for both placebo and for treatment. And so I think that you're exactly right. If there were more patients in the arms, this would probably be significant, but it would be significant to the same degree as obeticholic acid.
This is Jörn. I'm going to comment, and I think the trial obviously had that problem, if you'd like to say so, that one of the compounds used proved to be inefficient. And, then after having set up all your numbers and calculated everything, they even took out that selonsertib monotherapy arm in the end. I think that was mentioned. And that's, of course, one of the problems with that study. I think it'll add to the body of evidence with regards to fibrosis progression, which patients actually do reach cirrhosis in 48 weeks if you start out at F3. And to me, I thought it was a pretty severe patient population with regards to NASH activity. Ballooning was high. So I think that was a pretty good setup for a study to enroll your patients. Unfortunate that one of the compounds didn't read through in terms of efficacy.
Yep.
Yeah, Joern, you make a very good point, and I would point out that this exact same thing happened in a Gilead trial when simtuzumab, when they were doing their early phase 2A with selonsertib. Simtuzumab didn't work, and therefore it was kind of used as a placebo in a phase 2A with selonsertib. And now they've taken a step further, and they have the same kind of effect here. I'm not criticizing Gilead as much as saying that this is the type of thing that can easily happen when you're trying to do such an ambitious clinical trial with so many arms and 3 different compounds.
Yeah, let me add another thought to this, and that is, if you just took the presentation at face value, and you've been around long enough in the field of NASH drug development, you could go back to Rohit's original presentation of the selonsertib phase 2 data. And you saw something very similar with selonsertib versus placebo. Numerically greater benefit of selonsertib on fibrosis improvement, but not statistically significant. And then when that was taken into 2 800 patient phase 3 trials, both in F3s and then in F4s, the stellar trials, we know that it didn't work. So at face value, you could argue, well, this is something similar. And I would challenge that notion. The differences between this study and the one Rohit presented with selonsertib is the treasure trove of additional non-invasive and artificial intelligence data that we have to support movement of disease in the right direction. And the whole bile duct injury thing is just fascinating to me because, if you go back to the CymaBay data and the whole issue for halting that trial back in November, and then convening this liver specialist group of hepatopathologists, billy experts, and fatty liver experts, and then coming out of that meeting saying that these atypical findings actually were present at baseline biopsy, and then having their hold lifted by the FDA, speaks to this notion that there's more going on in the liver than just lobular inflammation, hepatocyte ballooning, steatosis, and fibrosis. And just in this year alone, we've now seen this notion of some cholestatic injury being present in NASH, and this study showing mitigation of that. And we've seen trials where GGTs have dropped remarkably, and we tend to link that in some way sometimes to, what you would expect with cholestasis and underlying oxidative stress. But I don't know, Peter, if you have a comment on those thoughts or what else we need to see.
Yeah, I would completely agree with you on ductular hyperplasia. I think it is a fascinating finding that really Van Adrian kind of identified initially, or along with other people. And the bile ducts, of course, can secrete a large amount of cytokines in response to proliferation. They're likely the site of the generation of hepatocellular carcinomas and with stem cells and so forth. And so what's going on with the bile duct cell in NASH in this proliferation, I think is, one of the important research frontiers. And I think some drugs that may inhibit that hyperplasia, such as integrin inhibitors, the alpha V beta 6 integrins and others, might be quite important in NASH progression.
So I think I want to make one comment from a statistical dimension, not a scientific dimension, which is that the reason getting power calculations right is so important is that you can see a lot of things happen, and in any kind of statistical process. And until you know what you're looking for and you can isolate on a population where you can identify it, or throw your sample sizes large enough that it's going to occur in a large enough subsegment of patients, a lot of these ideas become fascinating, and they even become scientifically plausible, but then they need to get developed. So I would hope that what we will see is either designs that focus on these issues and therefore can be calculated only for patients where you have reason to believe you're going to find that issue, or larger, more broader population-based trials. And now I do go back to your board member, Donna, who said, you need longer, larger trials to figure out some of the underlying dynamics of what's really going on here. And when these kinds of hypotheses emerge, I think they're fascinating. I think they're important. Now we need to find a way to get significant numbers behind them, because what power calculations are ultimately allow you to do is to figure out what really matters when it matters.
Yeah, Roger, that's an excellent point, and I would just go back to what Stephen said. If you go back to the simtuzumab selonsertib phase 2. Not only did the presentations look like they were the identical color scheme, it almost looks like a photocopy. And the only difference is that there were even fewer patients per arm in that selonsertib phase 2, probably half. So they did double it, but they still didn't get kind of enough numbers with all the groups that they have to show a significant difference with the type of changes that they have.
So, Peter, on the first episode of this podcast, one of the smartest guys I know said, and I quote, "Hope is not a strategy." And one of the ways that you execute hope as a strategy and then get it wrong is to believe too fervently in what you can do with smaller power calculations.
You're exactly right, Roger. And whoever said that is really quite brilliant.
For those who haven't been listening for the last 23 weeks, that was a corporate guy named Peter Traber. And I couldn't agree with you more. This is one of those topics where that really plays out big time. With that, why don't we move on to Stephen, who will be talking about the paper that he presented at 6 o'clock this morning, Texas time.
Hey, thanks, Roger. So this was the late breaker oral presentation that kicked off the late breaker session. So I think it's late breaker 1. Positive top-line results from a 24-week randomized, double-blind, placebo-controlled, multi-center phase 2 study of the FGF19 analog aldafermin, previously known as NGM282, in patients with NASH with moderate to severe liver disease, so F2, F3. And this is kind of a continuum of a large body of work that NGM is assimilating in the phase 2 space. If you're familiar with drug development, usually you do preclinical work, you do SAD/MAD studies in phase 1, you jump to a 2A in the field of NASH where you show some non-invasive data, you do a paired liver biopsy phase 2B trial, and then you're into phase 3. That's how all the phase 3s that are currently underway have been developed. I believe so. I may be off on one, but I think that's the classical route of progression. With aldafermin, it's been a bit more methodical. What we're reporting on today, what I reported on today, is cohort 4 of kind of an ongoing phase 2 program. Originally started with just a non-invasive assessment of 3 milligram versus 6 milligram treated for 12 weeks. That data was published in Lancet back in 2018. Because of the rapid and sustained drop in ALT and liver fat content, the cowboy in me decided to push for a 12-week paired liver biopsy study with 1 milligram and 3 milligrams of the drug. And there was no blinding. It was all open label because I did feel like if you were going to have liver biopsies 12 weeks apart, they should get the drug. And that showed some pretty robust results that I'll highlight at the end in comparison. That was subsequently published. And then now moving into this study, cohort 4. So the study design for this was a 24-week trial where you had to have a biopsy to get in and a biopsy to get out 6 months later. It was only 1 milligram of aldafermin given sub-Q daily versus placebo. So just one arm of the study versus one arm of drug versus placebo. Ultimately, there were 78 patients included in the safety population and 77 in the efficacy population. It was a 2-to-1 randomization ratio. So basically roughly 50 versus 25 patients. Primary endpoint was change in absolute liver fat content, not at week 12, but at week 24, with exploratory and secondary endpoints, including liver histology. So everybody got a liver biopsy, placebo and drug. You may be familiar with the Acaro trial that recently finished. Those that had a response in PDFF underwent liver biopsy. So not to confuse that, in this trial, everybody had a liver biopsy. And then we know, because of the mechanism of FGF19 inhibiting CYP7A1 and the conversion of cholesterol to bile acids, that we're going to see a rise in LDL cholesterol. That was known, and a mitigation strategy was implemented per protocol at the beginning assess the LDL at week 2. If it's elevated above 10 milligrams from baseline, then start rosuvastatin 20 milligrams. Continue to assess, and if you need to titrate up in your rosuvastatin dose, that was okay as well. The patients were evenly distributed between placebo and drug, about 60. And we see this over and over again. These more advanced studies of F2, F3, even F4s are predominantly diabetic. So here, 64% were diabetic. Liver fat content was pretty robust at around 18% by MRI-PDFF. And Pro-C3 levels were pretty high, around 17, with normal being around 10. And Pro-C3 is a marker of fibrogenesis. So just jumping to the chase here, the primary endpoint was met. That's the change in liver fat content in 24 weeks. It was an absolute reduction of 8% in the treated group versus 4% in the drug group, or in the placebo group. Maybe more importantly, about 70% of the aldafermin patients hit that 5% absolute reduction in liver fat content. About 66% achieved a 30% reduction in liver fat content. Just moving on to some of the secondary analyses that I think were important that I presented this morning, looking at fibrosis improvement of at least one stage with no worsening of NASH. Was a placebo-adjusted response rate of 20%, 38% for aldafermin, 18% for placebo. NASH resolution without worsening of fibrosis, 24% versus 9%. Neither one of those values were statistically significant, weren't powered for that, but what did hit statistical significance, really to my surprise, was when you combine both of those endpoints in more of a stringent endpoint analysis of both fibrosis improvement and resolution of NASH. It was 22% with aldafermin, 0 for the drug-treated group. We did not report today on a 2-point improvement in fibrosis, but we did do a deeper dive into looking at non-invasive testing to see if that could predict the probability of fibrosis improvement. And so we showed a slide, and maybe this is Joern's question earlier that we didn't get to discuss. We showed a slide where we talked about fibrosis improvement of at least one stage with no worsening of NASH at week 24, just in the F3 population. And it was 30% of patients in the aldafermin and 0 in placebo that hit that mark. But if you looked at stage 3 patients that had a liver fat content reduction of at least 30%, in other words, what we call a PDFF responder, then the percentage of people having at least one stage improvement in fibrosis, if you were F3, went to 46% versus 0 for placebo. And this has implications because, if you're able to look at PDFF along the way as a non-invasive marker, and particularly if you're F3, and then you can say, hey, look, we hit this magic number. Maybe we're at 35% reduction. We know we have almost a 1 in 2 chance that our patients are going to get some degree of fibrosis improvement. Now, this is small numbers, but at least it's interesting data. And it was substantiated actually by Rohit Lumba's presentation of resmetirom yesterday on an analysis, a further analysis of the phase 2 data with resmetirom, specifically looking at PDFF. And he showed very similar data that when you get above this 30% reduction in PDFF, there was actually fibrosis improvement that was statistically significant relative to placebo with a THR beta class of drug as well. So here's a different class of drug, completely different mechanism, both defat the liver as noted by PDFF change, and now you're seeing that having an association, I'm not saying correlation, but an association with fibrosis improvement. And then of course, there was improvement in the NAFLD activity score across the spectrum of steatosis, inflammation, and ballooning. ALTs, again, significant and sustained reductions were noted. And then there's the comment about the lipids. And what I showed was that specifically looking at the 10-year ASCVD risk score, that's a well-validated risk score. At baseline, the numbers were relatively similar. 12% mean ASCVD risk score in placebo, 15% in drug treated. The reduction was seen in both placebo and the drug treated arm over 24 weeks, but it was more accentuated in the aldafermin arm. The reason for that likely is only about a third of people in placebo wound up being on a statin, whereas almost 100% of the drug treated group did. But the surprising data that came out of this was what we learned at baseline. At baseline, 61% of NASH subjects had diabetes and otherwise qualified for a statin use based on the ASCVD risk calculator, but only 32% were actually on a statin, suggesting that these NASH patients, particularly the diabetics, we are underutilizing statin therapy anyway. And so ultimately, moving to safety and tolerability, interestingly, there was no difference in GI side effects between placebo and the 1 milligram of aldafermin. And looking back to the other data sets, 1 milligram is generally well tolerated. There was no increase in pruritus with this drug versus placebo. There were 2 serious adverse events in the drug-treated arm. Both were deemed not related to treatment by the site investigator. One was rectal bleeding. I got asked about that this morning. I don't have a lot of detail. I don't have any detail on that particular case, if it was hemorrhoids or if it were something more significant. It was just the PI felt like it wasn't related to drug. And then the other one was a liver biopsy procedure-related complication. So ultimately, just to wrap up, the study met its primary endpoint. There were clinically significant improvements in histology regulatory endpoints, even the more stringent endpoint of both NASH resolution and fibrosis improvement. Was very favorable safety and tolerability profile. The most common adverse events occurred were very similar in frequency in placebo and the aldafermin arm. So I think it bodes well for what we're potentially going to see in the next phase 2b trial that is currently enrolling, where patients are being treated up to 3 milligrams for 24 weeks. And so anxious to see how that 3 milligram dose stacks up to the 1 milligram that we saw today.
Okay, thank you, Stephen.
Yeah, this is Jörn. I'd like to comment, and I think it was a great presentation, Stephen. Congrats again on that. And I think I have 2 major things that I took away. One is the underuse of statins, and I can confirm that that's the case in most of my patients I've seen. And I think there's still that fear of liver tox in that NAFLD patient, that's been discussed previously. And I really think that's an issue we have to tackle in terms of, managing our NAFLD patients optimal before enrolling them into a clinical trial. I think that's crucial to not end up with an uncontrolled population that hits a trial and then we don't know what comes out. So I think that's important, and congrats to you guys to, study that so explicitly and actually manage the lipids actively. And the second thing I found very interesting was your comment on the combined endpoint that was kind of an exploratory thing and came up post hoc, and you actually saw significant improvements here. And I know you guys discussed AI on previous webcasts here, and I think that's where the power of machine learning comes in. And if you use additional parameters, not only imaging and histology, if you throw in something like ProC3 and some changes of blood markers that we might know today or not, I think we'll end up with a very good feeling of which patient responds how to which drug. I think we need that combined analysis of endpoints.
That's great points, Jörn, for sure. And just to tell you where I'm hoping we go with this data, and not just this data, but every other data that has robust outcomes. I really want to begin to focus on the 2-point improvement or 2-stage improvement in fibrosis. And I really want to begin to link magnitude of effect in some of these fibrosis biomarkers, either validating what Gilead has shown with ELF of a 0.5 drop in ELF. I we don't have anything in Pro-C3. What's an important number? What are we trying to hit with Pro-C3 that we can take home, knowing that that's likely to be associated with a fibrosis improvement? Interestingly, there was a poster that we also presented from this data on Pro-CX. I'm not sure if you saw that, but X is the cross-linking of collagen. And we had a poster looking at NASH and PSC, noting that the Pro-C3 X values were significantly higher in PSC patients, suggesting that there's quite a bit more cross-linking of collagen fibers in PSC or cholestatic liver injury. And I wonder, hypothesis generating here, is that one of the reasons why PSC is so hard to treat? Those cross-linked fibers may be more challenging to unravel than maybe a NASH fibrotic, for instance. So anyway, more to come there. But again, we've talked about Pro-C3, we've talked about Pro-C3/3CM ratio, which is looking at fibrogenesis and then fibrosis degradation, kind of a composite index, if you will. And now there's Pro-CX, which is interesting.
Jörn, why don't you go ahead, and then we'll see what we've got for time after that.
Okay, great. Well, thanks. Today, my abstract that I choose was late-breaking poster number 1. It was entitled Multifactorial Effects of AXA 1125 and AXA 1957 observed on markers of metabolism, inflammation, and fibrosis, a 16-week randomized placebo-controlled study in NAFLD with and without type 2 diabetes. Now, I choose this poster because it addresses a novel mechanism of action to me. It focuses on a mixture of amino acids, and the company calls them en masse endogenous metabolic modulators. I think that's a good name. It's a little bit difficult to grab what's behind it. In the end, it's a mixture of amino acids and different small molecules, for example, N-acetylcysteine, which we know is very potent, at least in animal models, to modulate non-alcoholic fatty liver disease. And going to PubMed and looking at some of those, I think there is actually quite some interest in studying different amino acids and their role in the disease pathophysiology. So I think those compounds have a pretty solid preclinical rationale to explore these targets. And actually a recent article from Anime Deal's lab studied glutaminolysis and its role on NASH fibrosis. So I think there's a lot of truth in those pathways, and it's an emergent mechanism of action that I thought was very interesting. So the study goal of this phase 2A non-IND trial was to explore the safety and the biological activity in a patient population with non-alcoholic fatty liver disease. It was not designed to show superiority of one or the other arms, and I'm going to address that a little later. But the study took a very smart approach from my side to include patients. It defined NAFLD by elevated MRI-PDFF above 10%. We've been seeing some different cutoffs here. Some studies do the 8%. This one used 10% cutoff. It also required and corrected CT1 of above 830 milliseconds. That means you enrich your patient population for more severe inflammatory NASH phenotype. And also fasting AST above 20 was required, and that kind of tips your population towards a little bit of fibrosis. No histology, so obviously a combined non-invasive tests and enrollment strategy, which I like, because coming from the clinic, I talk to my patients about an experimental drug. I think it's important to avoid additional harm, so those non-invasive tests for sure at the front runner with my patients, also from my side. So overall, a smart way to enrich your patient population. The study design was a randomized placebo-controlled trial. It consisted of 4 arms, the AXA 1125, 2 doses of AXA 1195, and actually placebo over a total time of 16 weeks. And I'm going to move on to the key findings now. So the primary presented endpoint is the relative change in MRI-PDFF. Remember, it was required to be elevated at inclusion, and the AXA 1125 achieved a 20.7% mean change from baseline at week 8, and that was maintained at week 16. And the placebo rate here was minus 5.9, minus 5.7 at the 8 and 16 week time points. So clearly numerically lower. The second compound that was studied in 2 doses, the AXA 1957, didn't quite achieve the same number here. And the second thing they examined is actually a change in HOMA-IR. And to me, that's very appealing because the AXA 1125 showed a decrease in HOMA-IR, indicating increased insulin sensitivity in patients that were treated with this compound, this modulator. And that wasn't seen in the placebo or the other low and high dose combination. So, decreasing liver fat has some meaning for our patients, in my point. And if you show that decreasing liver fat improves your metabolism, and that's beneficial even beyond the liver at many levels, I think this is really something to be on the lookout for. And not all phase 3 studies that are studying liver fat content and reductions are showing actually an improvement in HOMA-IR. So next, additional markers of disease activity were explored. Pro-C3, we've discussed this previously. And there's also a pretty robust reduction between at week 8, 1 and 2 points, absolute numbers, Pro-C3 reduction, and actually an increasing efficacy at week 16 with a reduction of between 3 and 4 versus 0.7 in placebo. So I like that because it tells me that fibrogenesis, which is measured by Pro-C3, is slowly turned off. It's not like you have the drug hit your patient and everything stops in the liver, but you see an incremental effect over time, and fibrogenesis is slowly reverting. That's how I see that early data here. You got to remember this is not an efficacy-powered study, but I like that signal that you get out of that Pro-C3 change, which is slowly over time. And actually did a physical health study where we saw a similar effect. We saw, delayed decrease in Pro-C3, and I think that's a good thing for patients. So other markers like ALT, the change in CT1, and also CK18M65 decreased predominantly in the AXA 1125, not so explicitly in the AXA 1957. The decrease in ALT was actually over that magic threshold of 17 in the AXA 125 at both week 8 and week 16. So that's also indicative of being able to observe changes on histology in the long run. So what else? I think we've seen in that Congress and during this Congress, this meeting and recent drug trials, we've seen that safety is so important. And of course, this trial was predominantly looking at safety of those 2 compounds. And it's good to say that it had a very convincing safety profile. Predominant AEs that emerged were diarrhea and nausea with the AXA 1125 dosage, and but only 1 discontinuation in the AXA 1125 related to upper abdominal pain. And I think that overall safety profile was convincing to me. Now, the diarrhea aspect is actually very interesting because there's a recent paper in the Lancet GastroHap on lubiprostone where that motility agent actually showed an improvement in ALT2, and I think it could even be a good effect if you get some gut motility side effects to address some aspects of those disease. In the end, I'd like to summarize and conclude on that paper that this is a non-IND trial that was designed to look at safety and some early efficacy data, but it wasn't designed to show superiority on one or the other. I kind of like the approach that it addresses multiple pathways that are involved in the pathophysiology in NASH. In German, we call it a Gießkannen concept. Maybe it's like your watering pan. You use a little bit of water on everything: fibrosis, inflammation, metabolic changes. And I think in the end, it could be an adjunctive medicine for patients. And I think there are plans to take this ahead, at least for the AXA 1125. So thanks.
Questions for Jörn?
Jörn, real quick, I thought you did an excellent job of summarizing the presentation. The interesting thing with this, having enrolled several patients in this trial, it's a powder that you mix with water, and it tastes like they say it tastes like Tang instant breakfast drink, the old Tang like the astronauts drank, which I think is, it's interesting. To me, I never would have imagined that a composition of various amino acids would ultimately have such a pleiotropic effect, or at least a broad-ranging effect on multiple different biomarkers. And I know in the presentation, we talk a little bit about the combination of hitting multiples of those, right? Hitting a 40-second improvement in CT1, 40 millisecond improvement, a 30% relative reduction, and a 17 unit per liter reduction. I'd be interested in your thoughts. Do you think additive combinations of important metrics for biomarkers lead to enhanced efficacy, or are we just fooling ourselves in that thought?
That's a very good point, and a difficult question to answer, obviously. From what I understand of the disease, looking at animal models, looking at the natural progression, I think it's important to address all these different pathways. And that's why I think this could be an exceptional concept as an adjunctive therapy to, either another drug or a patient that's really in a weight loss program and needs that little bit of kick to tune down his NASH and be on an antifibrotic pathage to start out. And then by losing weight, he'll, more easily cure his disease. So I think that's exactly what I like about this compound.
Peter?
I applaud this company for their approach and for the data, which surprised me in terms of the breadth of the result. And I just have a little unsettled feeling about what are essentially the nutritional combinations and what the actual underlying mechanism is. And I always wonder about gut-liver interactions and what's going on in the gut that might affect the liver, and how, different ratios of amino acids may affect, the gut microbiome and other aspects of the gut. So I don't know. It's just a nagging feeling when I look at agents such as this.
Peter, and I agree. You could consider it a nutritional supplement too, if you'd like. This is tested in the pharma field here, and they do have some evidence on which mechanisms are addressed. The combination makes it difficult to then dissect which compound within that mixture actually does give you the benefit. So I think that's an important question they have to look at too. But as I mentioned in the beginning, some animal studies explore that, and also the lubiprostone study that showed some effects with the GI motility. I'm on your side, and I agree. I think that could be parts of the effect we're seeing here. Some changes in gut microbiota, gut-liver axis.
Yeah, and I'm not, maybe it's just my problem in thinking through these type of compounds, but also metabolism is such that, and there are some amino acid metabolites in some of their mixtures, and of course that could shift as it, goes through the GI tract as well. But anyway, I think it's one of the more intriguing approaches in the field today.
I agree, Peter. When I first heard about this last fall, I guess, I had a hard time wrapping my mind around it because it was different, and I also thought it was tremendously intriguing. I believe because it's different, we're going to need to see more data for some of us to get comfortable with it, but everything they've shown so far would support what they say they can do. So I think that will be interesting to watch.
And I would say that they did comment at the end, and I think even Joern commented that they do plan on moving forward, at least with the AXO 1125, into an IND-enabled clinical trial. So I think you're going to get what you want.
Look, I think it's an exciting company. I can't wait to see more. Donna, first of all, had something she wanted to share with us.
Thank you, Roger. So as we have heard across this EASL meeting, the evolution in non-invasive testing, non-invasive diagnostics is accelerating and is needed more than ever, needed more urgently. And so the Global Liver Institute has had a Beyond the Biopsy campaign to shine that spotlight on the innovations in non-invasive technologies, particularly in NASH, but also in liver diseases as a whole. And so I'm very excited that we're going to be launching that campaign, which will go throughout the month of September with a radio media tour with myself and Dr. Mazen Nuradin, who is no stranger to anybody in the field of NASH. And so we'll be talking to radio stations across the country on Monday. And then you'll see a series of GLI Live episodes with folks also who need no introduction, like Dr. Scott Friedman and Arun Sanyal. So I hope that you will check out some of those resources and some of the ideas and things that we discuss as part of the Beyond the Biopsy campaign launch.
Donna, that's a really important and exciting initiative. Delighted to hear that you're undertaking it. I don't think it could be better timed based on some of the things we've been talking about and everybody's been learning the last 3 days. And I expect we'll learn more of the same when AASLD goes digital in a couple of months. So good luck with it, and congrats on doing it.
Thank you.
To our listeners, in the last 3 days, we've put down about 5 hours of recordings where we thought originally we were going to do 2.5 or 3. So we're going to do a couple things. First of all, instead of closing out today's session with comments from everybody, we're going to come back to this on Monday. Joern, I will drop you a note and see how we can bring you back into some kind of wrap up on Monday. Share some closing thoughts about the meeting with us. But Monday will be a shorter session. It'll be a session with closing thoughts about the meeting in general. We also are going to index the sessions that we've done so far to talk about the papers that we started, presentations we started, and at what point in the tape on which day we started to discuss each. So if you have something you want a listener go back to or missed or remembered, or you didn't have time to catch originally, we will be able to tell you exactly where on the file it is. That should be in place by, I'm going to guess, Tuesday or Wednesday of next week. And with that, we will be back on Monday. I want to thank Peter, Donna, Louise, Stephen. You folks have been heroic and energetic. Joern, thank you for being a spectacularly good guest today. And then I want to thank the production team, Tom McDonald, who stepped in, doesn't normally do this. And we've thrown more curveballs at him than a major league batter would see in the course of a season in the majors. Eric Rounds, Polly, Ryan, and you listeners who've made this a real joy to do by stepping in and listening so often and so quickly and responding so well. Everybody, enjoy the rest of your weekend. Those of us who've been getting up at some really dark early hour or staying up way late tonight, Joern, we're going to rest or relax in some other way. Everybody stay healthy. Everybody stay safe. Everybody surf on. We'll see you on the podcast on Monday. And thanks again for all of you. Bye-bye now.